Hello everyone. Today we discuss the biological databases. Databases are nothing but the collection of information. In case of biological databases, biological information is collected. So current biological databases use all three types of database structures, that is flat files, relational and object-oriented system. Despite the obvious drawback of using flat files in database management, many biological databases still use this format. So most of the biological databases are using flat file system. The justification for that is that this system involves minimum amount of database design and the search output can be easily understood by working biologists. Introduction to biological databases. Based on their content, biological databases can be roughly divided into three categories: primary databases, secondary databases, and specialized databases. Primary databases contain original biological data. So huge amount of original biological data is present in primary databases. They are archives of raw sequence or structural data submitted by the scientific community. Primary databases consist of the raw sequences and structural data system. Gene bank and protein data bank are example of primary databases. Secondary databases contain computationally processed or manually curated information based on original information from primary databases. Translated protein sequence databases contain functional annotation belong to this category. Examples are SwissProt and Protein Information Resources, that is PIR. Specialized databases are those that cater to a particular research interest. For example, FlyBase, which consists of the information about the flies, HIV sequence database, which consists of information about the HIV sequence, and a ribosomal database project, that are databases that specialize in a particular organism or a particular type of data. Primary databases. There are three major public sequence databases that store raw nucleic acid sequence data produced from submitted by researchers worldwide. GenBank, the European Molecular Biology Laboratory, that is EMBL database, and the DNA Data Bank of Japan, that is DDBJ, which all are freely available on the internet. Primary databases are freely available on internet. An example is GenBank, EMBL, DDBJ. Most of the data in the database are contributed directly by the author with a minimal level of annotation. So annotation is less in case of primary databases, and that data is submitted by author or scientist. A small number of sequences, especially those published in 1980s, were entered manually from the published literature by database management staff. So the sequence which is present before 1980 was manually inserted in that database by database management staff. Presently, sequence submission to either GenBank, EMBL, or DDBJ is a precondition for publication in most scientific journals to ensure the fundamental molecular data to be made freely available. These three public databases closely collaborated and exchange new data daily. So data which is present in one database is exchanged with the other database. They together constitute the International Nucleotide Sequence Database collaboration. So they can create a collaboration and they can exchange their data on daily basis. This means that the connecting to any one of the three databases, one should have access to the same nucleotide sequence data. Although three databases all contain the same sets of raw data, each of the individual databases has a slightly different kind of format to represent the data. So each database uses a different kind of format to represent the data. Fortunately, for three-dimensional structures of the biological macromolecule, there is only one centralized database, that is PDB. This database are through atomic coordination of the macromolecules, both proteins and nucleic acid, determined by X-ray crystallography and NMR. So data structural data which is submitted in PDB is based on which is determined by the X-ray crystallography and NMR. It uses a flat file format to represent protein name, authors, experimental detail, secondary structure, cofactors, and atomic coordinates. The web interface of PDB also provides viewing tool for simple image manipulation. Secondary databases. The sequence annotation information in the primary database is often minimal. To turn raw sequence information into more sophisticated biological knowledge, much post-processing of the sequence information is needed. This begs the need for secondary database, which contain computationally processed sequence information derived from the primary database. Secondary database consists of the computational information. The amount of the computational processing work varies greatly among the secondary database. Some are simple archives of translated sequence data from identified open reading frame in DNA, whereas others provide additional annotation and information related to higher level of information regarding the structure and function. Much of this information is obtained from scientific literature and entered by database curators. The annotation provides significant added value to each original sequence record. The data record also provide across referencing links to other online resources of interest. Other features such as very low redundancy and high level of integration with other primary and secondary databases makes SwissProt a very popular among biologists. A recent effort to combine SwissProt, Trimble, and PIR lead to the creation of UniProt database, which has larger coverage than any one of the three databases, while at the same time maintain the original SwissProt feature of low redundancy, cross-references, and a high quality of annotation. There are also secondary databases that relate to protein family classification according to function or structures. The PFAM and block databases contain aligned protein sequence information as well as derived motifs and patterns, which can be used for classification of protein families and interface of protein functions. So it consists of information about the protein families and interference of protein functions. The DALI is a protein secondary structure database that is vital for protein structure classification and threading analysis to identify distinct evolutionary relationship among the proteins. Specialized databases. Specialized databases normally serve a specific research community or focus on a particular organism. So it consists of limited information about a particular type of organism or particular information. The content of these database may be sequences or other type of information. The sequences in these databases may overlap with the primary database, but may also have a new data submitted directly by authors. So authors can directly submit the data in these specialized databases. Because they are often curated by experts in the field, they may have unique organization and addition annotations associated with the sequences. Many genome databases that are taxonomic specific fall within this category. Example include Flybase, Wormbase, S database, and TIAR database. In addition, there are also specialized databases that contain original data derived from functional analysis. For example, GenBank EST database and microarray gene expression databases at the European Bioinformatics Institute, that is EBI, are some of the gene expression databases available. So EST and GenBank EST database and microgene expression database are examples of specialized databases. There is an interconnection between the biological databases. Primary databases are central repositories and distributors of raw sequence and structure information. Primary databases consist of information raw sequences, information about the raw sequences and structure, and that information is used by secondary and specialized databases. They support nearly all other types of biological databases in a way akin to the Associated Press, providing new feed to local news media, which then tailor the news to suit their own particular needs. Therefore, in biological community, there is a frequent need for the secondary and specialized databases to connect to primary databases and keep uploading sequence information. In addition, a user often needs to get information from both primary and secondary databases to complete a task because information in a single database is often insufficient. Instead of letting user visiting multiple databases, it is convenient for entry in a database or to be cross-referenced and linked to a related entry in other databases that contain additional information. All these create a demand for linking different databases. The main barrier to linking different biological databases is format incompatibility. Current biological databases utilize all three types of database structures, that is flat files, relational, and object-oriented. The heterogeneous databases structures limit communication between databases. One solution to networking the database is to use a specification language called Common Object Request Broker Architecture, that is COBRA, which allows database program at different locations to communicate in a network through an interface broker without having to understand each other's database structure. It works in a way similar to hypertext markup language, that is HTML, for web pages, labeling database entries using a set of common tags. Similar protocols called Extensible Markup Language XML also help in bridging database. In this format, each biological record is broken down into small basic components that are labeled with hierarchical nesting of tags. This database structure significantly improves the distribution and exchange of complex sequence annotation between the databases. Recently, a specialized protocol for bioinformatics data exchange has been developed. It is distributed annotation system, which allow one computer to contact multiple servers and retrieve dispersed sequence annotation information related to a particular sequence and integrate the result into a single combined report. There are certain pitfalls of biological databases. There are certain drawbacks of these biological databases. One of the problem associated with biological database is over-reliance on sequence information and related annotations without understanding the reliability of the information. What is often ignored is the fact that there are many errors in sequence databases. There are also high levels of redundancy in primary sequence databases. Redundancy is nothing but the repeatedness. Annotation of gene can also occasionally be false or incomplete. Annotation means additional information. Annotation of gene can also associated be false or incomplete. All these types of error can be passed on other databases, causing propagation of errors. Most errors in nucleotide sequences are caused by sequencing errors. Some of these errors cause frame shift that make full gene identification difficult or protein translation impossible. Sometimes gene sequences are contaminated with sequence from cloning vectors. Generally speaking, errors are more common for sequences produced before 1990s. Sequence quality has been greatly improved since. Therefore, exceptional care should be taken when dealing with more dated sequences. Redundancy is another major problem affecting primary databases. Redundancy is repeatedness. There is a tremendous duplication of information in the databases for various reasons. The cause of redundancy include repeated submission of the identical or overlapping sequences by same or different authors, revision of annotation, dumping of express sequence tags, data, and poor database management that failed to detect the redundancy. This makes some primary database exclusively large and unwidely for information retrieval. Steps have been taken to reduce the redundancy. The National Center for Biotechnology Information, that is NCBI, has now created non-redundant database called REPSIC, in which identical sequence from the same organism and associated sequence fragment are merged into a single entry. Protein sequence derived from the same DNA sequences are explicitly linked as a related entries. Sequence variants from some same organism with very minor differences, which may well be caused by sequencing error, are treated as a distinctly related entries. This careful curated database can be considered a secondary database. As mentioned, Swiss Prot database also has minimal redundancy for protein sequence compared to most other databases. Another way to address this redundancy problem is to create sequence cluster database such as Unigene that collates EST sequence that are derived from the same gene. The other common problem is erroneous annotation. Often the same gene sequence is found under different names, resulting in multiple entries and confusion about the data. Our conversely unrelated genes bearing the same name are found in databases. To alleviate the problem of naming genes, re-annotation of genes and protein using a set of common control vocabulary to describe a gene or a protein is necessary. The goal is to provide a consistent and ambiguous naming system for all genes and proteins. A prominent example of such system is gene ontology. Some of the inconsistency in annotation could be caused by a genius disagreement between the researchers in fields. Other may result from imprudent assignment of protein functions by sequence submitters. There are also some errors that are simply caused by omission or mistakes in typing. Errors in annotation can be particularly damaging because the large majority of new sequences are assigned functions based on similarity with sequences in databases that are already annotated. Therefore, a wrong annotation can be easily transferred to all similar genes in the entire database. It is possible that some of these errors can be corrected at the informatics level by studying the protein domains and families. However, other eventually have to be corrected using experimental work. So that is all about the biological databases. Thank you.